Listo.
Okay, hello everybody. Sorry, I had a small problem, but I solved it now. So it's my pleasure to introduce today's speaker, Dr. Jean-Michel Anet. Dr. Jean-Michel Anet studied his PhD at the University of Toulouse in France, having Jean Denarier and Charles Rosenberg as advisors, and he graduated in 2002. From 2002 to 2004, he was a postdoctoral fellow at the University of California, Davis, in Doc Cook's laboratory. And in 2004, he joined the University of Wisconsin-Madison as an assistant professor. He has been there till 2004, and since 2014, he is full professor. So currently, Jean-Michel Anet is a professor in the departments of bacteriology and agronomy of the University of Wisconsin-Madison. His primary research interest is understanding the molecular mechanisms allowing efficient symbiotic associations between plants and microbes, and applying this knowledge to maximize the benefits of such association in agriculture. So I acknowledge Jean-Michel very much accepting my invitation to present this virtual seminar to the CCG community and colleagues from UNAM and other institutions. Perhaps you share with me that this is a very interesting topic, the topic of his seminar. I am glad to hear it again. And I also acknowledge Jean-Michel's interest in trying to perhaps in the future incorporate Mexican collaborators in this project that somehow began in Mexico, as we will hear in a few moments. So thanks, Jean-Michel, and let's listen to the seminar entitled Nitrogen Fixation on the Ariel Roots of Maize and Sorghum. So please write your questions in the chat, and we will have time for questions at the end of the seminar. So thank you, Jean-Michel. Go ahead.
Thank you, Gina, and all of you for the invitation to talk to you. And as Gina said, I'm really excited to present this work in particular because, as you will see, it originated in Mexico and is quite relevant, I think, to also Mexican agriculture. So I think I have decided for this seminar to I have a section on future directions and possible collaborations, which is longer than what I do usually for seminars. But that's really the goal, is to hopefully possibly develop connections and interactions. All right, so I'm going to talk about nitrogen fixation on the Ariel roots of sorghum and maize, which is something that we have been working on for a bit over 10 years now. So I'm going to start by pretty broadly by reminding you of the nitrogen cycle. Most plants, most crops get their nitrogen from the soil, mostly in the form of nitrates and ammonium. What we call nitrogen fixation is the conversion of atmospheric nitrogen into ammonium, and it can be done in different ways. It can be done through industrial nitrogen fixation, which is the Haber-Bosch process. And these days in agriculture, it's a large part of the nitrogen that crop receive is coming from industrial nitrogen fixation, from synthetic fertilizers. You also have atmospheric nitrogen fixation happening when you have lightning. It's more minor in terms of contribution to agriculture, but still happening. And today I'm going to talk a lot about biological nitrogen fixation. So biological nitrogen fixation is performed by bacteria and archaea. And some plants, you may be aware of the legume family, and I know many of you work on rhizobium legume association. Legumes are really good at associating with nitrogen-fixing bacteria, rhizobia. And that's why we have legumes in all our cropping systems around the world. And legume crops, as you know, need little to no nitrogen inputs. And on top of that, they can even provide nitrogen for subsequent crops through crop rotations. Unfortunately, crops outside of the legume family are not really good at associating with nitrogen-fixing bacteria, and in particular, cereals require large nitrogen inputs to actually achieve good yields. And so it has been a long dream of the community to improve nitrogen fixation in cereals in particular. So my talk is going to be very simple. I'm going to first talk about why should we care more about biological nitrogen fixation. Second, I will talk about the discovery of nitrogen fixation on Ariel roots of maize and sorghum. And then I'm going to talk about future directions. And as I said, I hope it may lead to get some of you interested in that kind of biological system. Coming back on the three different types of nitrogen fixations that I just described, atmospheric nitrogen fixation, as you can see, is relatively minor, less than 10 million tons of nitrogen fixed per year. Biological nitrogen fixation, again, bacteria, archaea on terrestrial systems contribute 90 to 140 million tons per year. And according to the latest numbers I was able to find, these days, industrial nitrogen fixation, or that Haber-Bosch process, contributes over 170 million tons per year of nitrogen. So clearly that industrial nitrogen fixation has an impact on the nitrogen cycle. It's a significant contributor to the nitrogen cycle now. It's even, as you see, even higher than biological nitrogen fixation on land. So first, that Haber-Bosch process is a good thing, and I'm thankful that we have the Haber-Bosch process. That process was invented at the beginning of the 20th century by two German scientists, but it is really during the Green Revolution that it's used, or the use of synthetic fertilizers increased. So on that graph, what we're showing is the increase in red of the total world population. And in blue, it represents the fraction of that population which is sustained, fed by industrially fixed nitrogen. These days, we estimate that about half of the world population is fed through synthetic fertilizers. And so obviously, I'm not going to criticize that. It's a good thing. We're feeding half of the world on nitrogen fertilizers. And fortunately, we have these fertilizers. But of course, there are some drawbacks and issues associated with the intensive use of fertilizers. So synthetic fertilizers, first, nitrogen fixation requires a lot of energy to break the triple bond of dinitrogen and to form ammonium. And in the Haber-Bosch process, that energy comes from natural gas. And so we use natural gas to produce synthetic fertilizers, and we use these fertilizers to grow our crops for food, feed, fiber, and biofuel production. So first, regarding food and feed, if you think that what I said before, that we are feeding half of the world through synthetic fertilizers, what that really means is that we are feeding half of the world through natural gas, which is a non-renewable resource. So these days, and especially because of fracking, natural gas is available, but it's still a non-renewable resource. So when you think about sustainability of food production in the long term, clearly that's an issue. When you think about bioenergy crops that we grow and where we use synthetic fertilizers, there it's almost ironic, it's almost funny that we're using natural gas, a non-renewable resource, to make fertilizers to grow bioenergy crops for biofuel. So there clearly it's not efficient. So besides these sustainability and efficiency issues of the system, there are also some issues that are even more short-term. First, in developing countries, their problem is getting food. And in many places of the world, as nicely reviewed in that review from 2012, access to nutrients and access to nitrogen in particular is limiting. And so in all the places where you have either red or blue, these are places where nutrients are limiting crop production. And here in that case, I think that figure is for corn, maize production. So the problem with nutrients or access to fertilizers in developing countries is, one is the cost, but what is even more frustrating is that even the logistics of bringing these fertilizers to poor growers is an issue. And both issues, the cost and the logistics aspect, are very well described and shown in that book, The Last Hunger Season, from Roger Thoreau. So it's a great book. I highly recommend it. It talks about Kenya and the situation of the problem of nitrogen in particular in Kenya, showing both aspects, both the cost and the logistics. And there are also, as you probably know, environmental issues, and nitrogen leaches in groundwater, leading to eutrophication of streams, degradation of coastal zones, And you probably have heard about the dead zone in the Gulf of Mexico, which is a direct consequence of all the fertilizer we use here in the US and the Midwest. And also, these fertilizers are degraded into greenhouse gases, and that contributes to global warming. And so these issues are everywhere in the world, but in particular in places in countries that use a lot of synthetic fertilizers. So for all these reasons, and for a long time, in fact, the scientific community realized that we need to rely a bit more on biological nitrogen fixation. And I'm not hoping or claiming that we are going to replace all the synthetic fertilizers with biological fixation, but just replace some of it to rely less on industrial fixation and try to reduce the issue that I mentioned before. So biological nitrogen fixation in bacteria and archaea is performed by an enzyme complex called the nitrogenase complex. And I'm not going to go on the assembly or the function of that complex. It has been studied for decades. I'm showing that figure mostly to show the issues or the challenges with biological nitrogen fixation. One, it's thermodynamics. Again, if you want to break the triple bonds of the dinitrogen to form ammonium, you need a lot of energy. And so for bacteria or archaea that want to fix nitrogen and use the nitrogenase complex, they need to have access to a large source of energy. And in many plant bacteria associations, that energy often comes in the form of sugars, right, of a carbon source to provide energy to be able to fuel the nitrogenase. Energy access is very often limiting for a nitrogen-fixing bacteria. And the second issue that you may be aware of is the oxygen issue. This complex, that nitrogenase complex, has iron cofactors that get oxidized really easily, and oxygen is a strong inhibitor of the nitrogenase activity. And so in all the systems where you have efficient nitrogen fixation, you need to have a low oxygen environment and a high carbon source for the bacteria. So it's a little bit of background for what I'm going to talk about later. So for a long time, as I said, the community realized that we need to do more on trying to to develop solutions to allow cereals in particular to have access to more biological nitrogen fixation. A very classical approach has been in the field to either isolate and try to put some nitrogen fixing bacteria isolated in nature on cereals, or more recently to engineer these diazotrophs to fix more nitrogen and release that nitrogen. So it's an approach, the pure isolation and putting bacteria isolated from the field onto crops, honestly, was not very successful and is leading to very little amounts of nitrogen delivered to the plant. The approach of engineering the diazotrophs, the bacteria, is more promising. I'm working on several bacteria that we are engineering. I'm also working with a company called Pivot Bio, and where that is also using that approach of engineering the diazotrophs. That approach is interesting. Another approach is really long-term, and it's to take that nitrogenase complex and put it into the plants. So somehow removing the need for plant microbe interactions and directly engineering the nitrogenase complex into plant. It's a great idea. Several people in the world are working on that. Again, very long-term because there are some huge challenges. A third approach, which is in fact, if you read the literature, the one that people thought about first, I even have a paper from 1917, where they are literally talking about that approach of engineering, taking the legume system that we know that is very efficient and putting that into cereals. Again, great idea, but lots of challenges. So my lab is also working on such an approach of engineering nodules in non-legumes, but again, it's genetic engineering and long-term. All these approaches that I'm showing here, they all involve transgenic approaches, and it involves modifying bacteria, modifying the plants, or modifying both of them together. The approach that I'm going to talk about today is different. And so the idea was, is there something that works in nature already? And exploring natural diversity, and in particular, natural diversity on the plant side, on the host. I wanted to show you that review from Eric Triplett. Some of you know Eric, and he was working, in fact, here in Madison for a long time. Now he moved to Florida. But in 1996, Eric Triplett wrote a review first he was stating that, or reminding to the community that how that the development of nitrogen fixation in maize can be considered a holy grail of nitrogen fixation research. In particular, in the 80s and early 90s, there was a strong focus on that. And one approach that he was suggesting in that review, he was saying later, it may be necessary to isolate maize diazotrophic endophytes from many locations, including the areas of maize origin in Mexico. So his idea there is about not going back to the place where maize was domesticated and trying to look for natural diversity, but he was thinking bacterial side. What I'm going to talk about, it's very similar, but except we did not look at the microbial side. We looked for that project at the plant side. Is there natural diversity on the plant regarding nitrogen fixation? And for that, let's go to the origin of the place where maize was domesticated. So that project was looking at maize diversity, and in particular in places where close to where the place of domestication and center of diversity. And so that's what I'm going to talk about. And the project really got started with a collaboration from growers from a village in Oaxaca. The village is called Totontepec, Vilas de Morelos. It's in the mountains of Oaxaca, in what is called Sierra Mije. The maize, so first it's in the mountains. It's altitude, you see 1,500 to 2,000 meters above sea level. And maize is grown here. So here, these are the fields. Maize is grown in monoculture, so it's and continuous maize on the slope of the mountain. And the maize is growing beautifully. And what is really striking is that people over there don't use any synthetic fertilizer to grow the maize. They grow it in a traditional way without synthetic fertilizers. And the soil nitrogen levels are extremely low. So seeing maize growing like that so beautifully under such conditions was surprising. Something else you need to know about Totontepec is that it's a very humid climate. Humidity above 80%, it rains several times a week, and temperature relatively constant during summer, 25 to 30 degrees. So very humid environment. The maize is surprising in many ways. First, it's a giant maize. Well, here, the two people that you see in the field, they are not children, they are adults. It's to give you the scale. These are giant maize varieties. They are about 5 meters high. So if you're used to classical hybrids that we grow, at least here in the Midwest, it's twice the size of what we call a tall corn. So it's pretty big, and it's producing beautiful ears, great, dread crop, and very surprising, at least by its size when you see it. So the first approach that we used to look at nitrogen fixation was a technique called 15N natural abundance. And that technique relies on the fact that in any soil, you have a heavy isotope of nitrogen called 15N, which is relatively abundant. Of course, it varies from place to place, and that's the challenge of the technique. But if you're comparing plants within the same environment, the 15N level is relatively stable. So you have 15N, that heavy isotope of nitrogen in the soil. In contrast, you have very little amount of 15N in the air. And that's the principle of the technique, is to use that. So if you have a plant that is not fixing nitrogen growing in the soil, the 15N content of the plant overall will be very similar to the 15N content of the plant. The 15N content of the plant will be very similar to the soil. Of course, you do have some segregation of 15N within the plant, but overall the content of the plant will be similar to the soil because the plant is getting all its nitrogen from the soil. In contrast, if you have a plant that can fix nitrogen, it's getting more nitrogen from the air, which is really 15N poor. And so what you observe is a decrease in 15N in plants that fix nitrogen. And that was the basis of our first assay. The reason we use that is that it's really easy. You just do leaf sampling or soil sampling, and you can do that without any massive intervention. And so that we used at the beginning with the corn from Totontepec, and we started getting first samplings in 2010, a bit more than 10 years ago. And at the time, honestly, we were not very hopeful. So we did in 2010 only one sampling. And we did that 3 months after planting. But surprisingly and shockingly, the result came that using that approach, it was suggesting that the maize was getting a half of its nitrogen from the air. So quite frankly, in 2010, we really did not really believe it. And we came back and we established a collaboration with families in Totontepec and came back to in 2011, 2012, and doing a much more detailed time course over the season. Before 2 months and after 6 months, we detect absolutely no nitrogen fixation. But starting in the second month a little bit, but mostly in over 4 months during the growing season, we were observing that using that technique, that about half of the nitrogen of the plant during these 4 months is coming from the air. So that was surprising. Still, to be honest, we were very skeptical. And any technique that is using, any technique to measure nitrogen fixation, in fact, has some pitfalls. And classical pitfall associated with that one is if you have a soil where you have very different nitrogen contents in the soil, in different layers of the soil. So there could be artifacts. And so that was honestly our state of mind in 2012, is to think, well, there must be something wrong. Well, before I go further, I need to talk about other features of the maize. And one of them is that maize has many nodes with Ariel roots. Here, these are my children, my kids, my children really love these roots. They go with me to the field. And so what you can see is that these maize varieties, they have many nodes with these roots. We call them Ariel roots. Some people call them brace roots. I call them Ariel roots because these ones never reach the ground. Brace roots are, you probably have seen maize with brace roots at the base that serve for anchorage of the plant, but these roots never reach the soil. So we call them Ariel roots. So many nodes with Ariel roots. And we quantified that. So if you take a regular maize, for that project, we use a variety called Hickory King because it's also a giant corn maize, and we wanted to have something comparable with the maize from Totontepec. But most maize varieties that you're going to find, they're going to have typically 2 or 3 nodes with Ariel root at the base. If you compare the maize from Totontepec, has typically 8 to 10 nodes. Some plants sometimes have 12 nodes with Ariel roots. So they're literally covered with roots. That arrow here indicates the transition between the juvenile phase and the adult stage. And in maize, we typically divide the growth stages of plants with juvenile, adult vegetative, and adult reproductive. The transition here between adult and juvenile, we have different ways to evaluate that. And most classical one is to look at the wax on the leaves. And if you look at the literature, what is reported widely is that these Ariel roots are a juvenile trait. Well, what we observe in the maize from Totontepec is that we observe the transition from adult to juvenile, from juvenile to adult about at the same time as regular maize, but these varieties keep producing roots even at the adult stage. And and that goes against what most people think about when they think about maize development. We are currently working on a paper to solidify that claim, but it's pretty clear. We even have some roots produced after tassening. So clearly these accessions keep producing Ariel roots at the adult stage, which is quite unusual. The other unusual thing about these roots is that when it rains, they produce a lot of gel. We call that a mucilage, and each root can produce up to like 1.52 milliliters of gel. I always show that video because I think it's pretty striking. Of if you go to the field after 30 minutes, 1 hour after rain, you literally see these plants dripping with gel on their roots. So that was a bit unusual. So as I said, regarding nitrogen fixation, we had the 15N natural abundance data, but we needed more evidence, different techniques to look at nitrogen fixation. And so we brought some plants here and looked at different plants, different parts, plant tissues. When we evaluate nitrogen fixation using the nitrogenase activity using the acetylene reduction assay, We see absolutely nothing happening in leaves, stems, or even Ariel roots before rain without mucilage. We always see, like any other maize variety, we always see a little bit going on in the underground root system, but it's really small. But in contrast, if we look at these Ariel roots covered with gel after rain, we were detecting a huge activity. If you remove that gel from the plant, you don't see any activity in the root where you remove the gel. But in contrast, if you just collect the gel and look at the nitrogenase activity in maize collected either on Totontepec, and that was sent to us directly here for our activity, or a maize collected, gel collected in Madison, we see a strong activity just in that mucilage. So what we can do also, what we did is that we took that gel, we used freezing and thawing steps to abolish the nitrogenase activity, and then we re-inoculated that gel with different bacteria. And so here it's that gel that has been again went through freezing and thawing, where we basically kill the endogenous nitrogen fixing bacteria. And so without any bacteria, we don't detect any nitrogenase activity in the mucilage. And when we add here, it's herbaspirillum or isospirillum, we get a very strong activity again, indicating that the mucilage is a good environment for nitrogen fixation. And if we put the same bacteria in just a regular medium, we don't see any nitrogen activity. So clearly the mucilage provides a good environment for nitrogen fixation. What is that good environment? Well, that comes back to my introduction about issues with the nitrogenase. One is oxygen. We think that the viscosity of the gel is really important, and that it in particular slows down oxygen diffusion. When we measure oxygen level in just control, that was just water, versus compared to the mucilage, in just water, you get about 20% oxygen, but in the mucilage, you get 4-5% oxygen, which is an environment which is suitable for a wide range of aerobic or microaerobic nitrogen fixing bacteria. And the second thing that we think it provides, at least, is sugar. And what we see is the mucilage is extremely rich in sugar, and in particular in, as you can see, galactose, fructose, with arabinoses here, arabinose. So we think that that mucilage provides, at least solves some of the problems that I mentioned before that are necessary to solve if you want to have efficient biological nitrogen fixation. The problem, as I mentioned, with the field of nitrogen fixation is that we don't have a perfect technique to evaluate nitrogen fixation. We have different techniques, and here I'm listing the 5 major ones. And all 5 of these techniques have potential pitfalls and issues. So in practice, basically my point here is don't rely on a paper that uses only one technique because it has pitfalls. And so typically in our field, people use a combination of techniques. Because the claim that we had about that maize was pretty big, we used all 5 of them. And so I'm not going to go through the data for all 5 of them. I would just refer to the paper that we published in 2018 in PLOS Biology. Everything, and it's public, it's open access. The question, of course, that you may have is the plant itself is not fixing nitrogen. So what are the bacteria doing that? So we used, and we're still using, amplicon sequencing, shotgun metagenomics, isolation of bacteria, nitrogen fixers, and also non-nitrogen fixers, and sequencing of these genomes. Briefly here, that PCA plot is showing the results of the shotgun metagenomics, in fact, for that one. And what we are showing is that the mucilage samples that are here in blue are very different from the samples from the other parts of the plant. So the mucilage clearly has selects a very special community. It's high sugar, low oxygen environment. And the third aspect I did not mention so far, it's also a low nitrogen environment, which was very surprising for us at the beginning, thinking that bacteria fix nitrogen in there, until we realized that that gel is literally sitting on the root, and the root, it pumps nitrogen very actively outside of the gel. So the root is taking out ammonium and even nitrate, which is in the gel. We showed that using labeling experiments. And so the gel is actually a low nitrogen environment. And so it's a very special environment with low nitrogen, low oxygen, and high sugar that clearly selects a very special community. And the second aspect is that community is highly enriched for nitrogen-fixing bacteria. So clearly, the ability of these maize accessions to produce mucilage is critical to the trait. And so we looked at mucilage production and what are the factors that are important for mucilage production. One, which is very clear, it's thickness of these Ariel roots. If you look at brace roots of typical maize varieties that we use, these brace roots, first, are not many of them, but also they are usually very often very thin. And here what we did is that in that experiment, so here it's example of some roots from these corn landraces, maize landraces, and here these are roots from a typical inbred that we use here. And what we did is that we looked at the correlation between root diameter, diameter of these Ariel roots, volume of gel that these roots produce, and nitrogenous activity. And what we can see is that we have a very strong correlation between all three parameters. When roots are thick, they produce more mucilage, and when they produce more mucilage, they can support more nitrogenous activity. So a lot of the trick, if you want, is to produce a lot of gel, a lot of mucilage, and that directly correlates with the nitrogenous activity. The second aspect is the age of the roots. When these Ariel roots develop, when they just emerge from the plant, that they have a very nice little root cap, and that's actually where they produce the most mucilage. So what we did is that we defined some developmental stages for these Ariel roots, with the young ones, what we call stage 1. Here you can see it's very tiny root, but it's literally filled that 2 ml tube with gel. And stage 2 produces some gel, but less. Stage 3, stage 4, almost no gel. So here, these are older roots. When the roots start to become darker, their root cap in particular starts to disappear. At stage 4, the root is dry. It starts to dry. So first, the diameter of the root is fairly stable, and stage 4, it's really when these roots start to dry. And we see in terms of volume of gel produced, the vast majority of the gel is produced by that kind of young, short roots that just emerge and that have a very intact root cap. I talk about the root cap a lot because we think that the mucilage production is basically the site of mucilage production is the root cap. Here we made sections, transversal sections of such Ariel roots and added water. And as you can see here, that's the section of the tip. And you can see a lot of mucilage around, and still some mucilage and less. So clearly, mucilage production is coming from the tip. If you see these plants in the field, you may sometimes have the feeling that it's coming from the base of the root. And the reason is that these plants also have different, on the same node, they have older roots and younger ones. And if you see a mucilage at the base of a big root, it's actually because behind it, you have a young emerging stage 1 Ariel root. So basically the gel is produced by the root cap of Ariel roots, and in particular by cells that are called border cells. And these cells are not unique to Ariel roots. And as you know, you have a root cap also in underground root systems, and border cells have been well characterized as being the cells that are detached from the root cap and produce the gel underground. Well, it's exactly the same in these Ariel roots. The gel is produced by the root cap, except that root cap is huge because you have thick roots, and these huge root caps release a large number of border cells. Again, looking at stage 1, stage 2, and stage 3 roots, we looked at the border cells. We use a stain, India ink, which is an ink that cannot penetrate in the gel. And so here you can see what you can see here. Well, first, what you can see here is the gel is full of border cells. In fact, if you come back even to my picture I had at the beginning of the talk, you can see literally a cloud of border cells close to the root. So the gel is full of border cells, and you see each border cells produces the gel. So you can visualize the gel by looking at the white halo around these cells. So here that's the gel, and the ink cannot diffuse inside of the gel. So it's a nice way to visualize gel production. And stage 1 roots produce many Ariel roots, and these roots produce a lot of gel. So first, in terms of number of border cells in the gel, we see many of them in stage 1 and stage 2 roots, nothing in stage 3 or more. So again, consistent with the fact that most of the gel is produced by these border cells at stage 1 and stage 2. Interestingly, the size of the border cells is also different. Stage 1 roots produce bigger, larger border cells. And these larger border cells produce even more mucilage, which can explain also why these young roots produce more. All right. The gel itself, its composition was published by colleagues at University of Georgia, and it's a polymer of galactose with fructose and arabinose, xylose, mannose, and glucuronic acid. We think that that gel, as I told you, is degraded into simple sugars, and that these simple sugars is degraded by the bacteria, but also possibly by plant enzymes, and that these simple sugars are then used by the microbe to fuel nitrogen fixation. As I told you before, so we think that the gel, because of its viscosity, the polymer is responsible, acts as a diffusion barrier for oxygen. It's possible that the bacteria also help in that process of reducing oxygen tension, but clearly the viscosity of the gel creates that low oxygen environment. And also the last point, which I think is really important, is that the gel is a low nitrogen environment because it's sitting on the root, and the root is actively pumping nitrogen out of the gel. By the way, coming back on my border cells, we can see that actually very well here. You can see that's the root, and I don't know if you can see the cloud here around my pointer. These are the border cells. As you can see, you have all the border cells are here, and here we have some gel that have fewer border cells here. So that's what we think is the function of the border of the gel. All right. So what I talked about today was a lot focused on what we did with the maize coming from one village, Totontepec. It turns out that the collaboration with Mars stopped in 2018. And the reason is that Mars tried to patent the trait, and fortunately, I would say, the patent was declined because it's a natural trait, and the company decided to stop funding that project. I decided to continue that project, and for that, I decided to collaborate with the CIMMYT. And many of you know the CIMMYT is the International Center for Research on Maize and Wheat. And in collaboration with the CIMMYT, we started screening accession coming from other places in Oaxaca. I only looked at accessions in Oaxaca, and I must say that's hint that that could be something interesting to do elsewhere. But looking at accessions from Oaxaca with the CIMMYT, we actually found accessions again from Totontepec having the trait, accessions from the region, not only Totontepec in the mountains, having the trait, but as you can see, you also have accession from the lowland of Oaxaca near the sea that also have clearly the trait. So the trait is not unique at all to the village of Totontepec, it's throughout the region. And again, I have not even explored further, but I would not be surprised if the trait is in accessions from other places. I'm very interested in natural diversity, but I'm also very interested in evolution. I think evolution makes every biological story just more interesting. So we looked not only at the maize from Totontepec, we also looked at Teosinte species. And in particular, what we use is a Teosinte called Teosinte mexicana. We use two Teosinte mexicana and Teosinte parviglumis. Teosinte parviglumis is the direct ancestor of most of the maize that we grow. Teosinte parviglumis doesn't have the trait that I just described. In contrast, Teosinte mexicana has, here it's a picture on the right, has many nodes with roots. These roots, however, are thin, and so they produce less mucilage than the roots of the maize that we're interested in. But still, the trait of many nodes with roots is clearly present in Teosinte mexicana. And more recently, we even looked at Teosinte guatemalensis, that also has a high number of nodes with roots. When we looked at nitrogenase activity in the gel produced by, at the time here at Teosinte mexicana, it basically shows that, yes, there is actually good nitrogenase activity in the gel produced by Teosinte mexicana. is the gel, and here we tested also the ability of the gel from Teosinte mexicana to limit oxygen diffusion, and so to provide a low oxygen environment. And I don't show you the data on sugar, but conclusion is that the gel produced by Teosinte mexicana provides oxygen protection and provides sugars very similar to what we observed in the maize, with the maize from Totontepec. So clearly, my point here is the trait in Teosinte mexicana, maybe not as good in terms of amount of gel produced, but a vast majority of the trait is already in Teosinte mexicana. And as you probably know, Teosinte mexicana is like a weed. It grows in nature and very close to maize. And people have reported many introgressions of Teosinte mexicana into domesticated maize. And in particular, I think about the work of Jeff Ross Ibarra at UC Davis. And so our hypothesis is that the nitrogen fixation trait probably came from introgressions from Teosinte mexicana, that the maize was domesticated as Teosinte parviglumis, and that very likely there has been these introgressions from Teosinte mexicana that brought the nitrogen fixation trait. We are still working on that evolutionary story, but I think in the context of natural diversity, it's quite interesting. I also wanted to show you that one because if you look, so here it's a drawing from Europeans that came to Mesoamerica in the 16th century, and they made these drawings of the corn that they were seeing cultivated by native people. And if you're looking at the corn that they drew a corn with a maize plant with many nodes with Ariel roots. And so clearly it's not a novel story. I mean, clearly maize with many nodes with roots has been there for a long, long time, and probably thousands of years. So I thought that historical perspective was interesting. The trait is present in many varieties of corn. It's present in, as I said, the progenitor of corn. So the trait was probably in Zea mace, in Teosinte, even before domestication. And so we wanted to look even further. And so we looked at sorghum. And the reason I got interested in sorghum in the first place is because we stumbled on this cover page of a conference that was held in India at the ICRISAT in 1986. It was a conference on cereal nitrogen fixation. And what it's showing here, it's a sorghum plant with Ariel roots dripping with gel. And here is a micrograph of azospirillum. Interestingly, when you look inside of that book of abstract, or if you look at the literature, there is absolutely nothing about nitrogen fixation on the Ariel roots and sorghum. So we contacted Dr. Wani, who actually put that picture on the cover page, and asked him why. It's in fact, he had actually some preliminary data on nitrogen fixation in these roots of sorghum, but did not pursue that research. And literally, he stopped that aspect in 1986. So I, in fact, contacted him even last year to discuss that more. And he said that he put that picture to stimulate interest of the community in pursuing that. But obviously, nobody followed up in 1986. So we followed up more recently with colleagues at the ICRISAT and started collaboration with colleagues at ICRISAT to screen sorghum accessions with the trait. And briefly, we found sorghum that has exactly the same trait. Many nodes with roots, gel produced by these roots after rain, and good nitrogenous activity. And so now we have a parallel project in sorghum. Sorghum is also a quite important crop in Mexico. So if you're interested in that aspect, we can talk. The trait is mostly, of course, the microbes do the nitrogen fixation aspect, but the trait is controlled by the plant, a lot controlled by the plant, by plant genetics. And so since the beginning of that project, in particular, the Mars company was interested in trying to see if we could introgress the trait into varieties adapted to the US and to the Midwest. And so these are experiments where here you have parents that are called XPVPs, that are commercial varieties, commercial inbreds. And here are the nitrogen fixing Tiramihe accessions. And they crossed Tiramihe accessions to these varieties and obtained progenies. The technique that we used here, it's called 15N dilution. It's just a variation of 15N natural abundance in which we enrich 15N in the soil. And so in that kind of experiment, just like in natural abundance, a decrease in 15N is indicative of a nitrogen fixation happening. So what you can see here, the parents, well, high 15N, they clearly don't fix nitrogen. The serum accessions show a clear decrease of 15N, even in that greenhouse experiment. And interestingly, in the progeny, the F1 progeny, you see a segregation of the trait, which is indicative of that some individuals in that progeny acquired the trait. And so that really means that we can breed for the trait, that we can do genetic for the trait. So the project was initiated by Mars, but as I said, Mars completely stopped. And so I'm not working with what Mars was doing. And as I said, I started working again with the CMIT. And so currently I have basically resumed this entire breeding aspect with lines available from the CMIT and also lines coming from a USDA collection called the USDA Green. And the project is currently, we are trying to get that project funded by USDA. So now we are basically doing breeding using lines from, again, USDA Green and the CMIT, our public collections, and our work is public and will stay public. I want to be clear with that. All right, so future directions. I wanted to talk a bit about what we are currently doing. First, the trait is awesome, but also we don't want to be too naive. We know that the trait, for instance, requires a good amount of water. It's coming from regions with high humidity and a lot of rain. So we want to see what are the requirements of the trait for water. How low can we be in terms of irrigation and rain and still have decent nitrogen fixation? We want to test, and we have started doing that, the effect of soil nutrients on nitrogen fixation, of course, nitrogen in the soil, but also phosphorus and potassium. And most importantly, we want to evaluate the cost of the trait. So we have these plants producing, literally exuding sugar on their roots. Does that have a cost in terms of yield? Is there what we call a yield penalty? Maybe yes, maybe not. Corn is a really good plant that's doing maize is doing a lot of photosynthesis, and so maybe the maize can compensate or not. And so that's what actually we are doing right now. We are trying to evaluate if there is a yield penalty. We also, of course, want to understand better the genetics of the system. We want to understand, I love that picture because you can see all the nodes. We want to understand the genetics of the high number of nodes with roots. As I told you, I think it really has to do with the ability of these plants to keep producing roots at the adult stage, which is really unusual. If you read the literature, these roots are only produced at the juvenile stage, but clearly it's not the case here. We want to look at the genetics of the diameter of the roots and, of course, about nitrogen fixation. I'm also quite interested in the evolution of the trait and trying to understand where is the trait coming from. Well, clearly it's coming from Teosinte mexicana and possibly other Teosinte species, but also to try to understand the impact of domestication. What has been the impact of local communities in maybe even further improving the trait. So that also is something that I'm super interested in, and for which also collaborations are welcome. Microbe, we are of course interested in the understanding what are the microbiome players, what are the nitrogen fixing bacteria, but not only. We know that inside of the gel you have a whole community, and so we keep doing metagenomics, isolation, and with the goal of creating small synthetic communities and possibly using these communities for inoculation of the plant. Something I did not tell you is that when we look at the plant in the field and do these ARA assays, only half of the roots have nitrogenous activity. Half of the roots have nothing. And in terms of gel composition, it's the same. And so we really think that the fact that some roots don't have nitrogen fixation is just because of the chance that nitrogen fixing bacteria are going to land on these roots. And so that means that I think there is a huge potential for basically inoculation there and adding the nitrogen fixing bacteria and improving nitrogen fixation even for the initial land races. So I think there is a huge potential for inoculation. Of course, we want to kind of try to combine projects within my lab. We also work on genetically modifying the nitrogen fixing bacteria, trying to see if that helps, but really with the goal of, can we develop inoculants? Can we improve this system even further by providing good nitrogen fixing bacteria and possibly others? And the last one is really of course, the breeding aspect, is introducing the trait into maize varieties. My focus for that project is really developing countries, and for two reasons. One is, first, it's a tropical trait, and it's coming from tropical regions. And so it's, in terms of how easy it's going to be to achieve the goal, I think it's going to be, the first easy goal will be to put that trait into maize for tropical regions. The second aspect is my concern about yield penalty. If there is a yield penalty, there is no chance that growers in like the Midwest of the United States are going to accept a maize having lower yield, even if it's fixing nitrogen. And in developing countries where people struggle to get nitrogen in the first place, well, yield is not necessarily the top priority. The priority is to have a corn growing under low to no nitrogen. And so I think that goal is more actually achievable, at least in the short term, than trying to develop maize for the US, because honestly, I think it's going to be challenging. So that's my point here, is that with, in particular, Claudia Calderon, who is my main collaborator here for the breeding aspect, we are really interested in doing participatory breeding and working to breed for, in particular, developing countries or places where they need nitrogen in their cropping systems. The goal of putting that into maize for the US, honestly, maybe, but super long term, and yeah, we'll see. All right, so that's my acknowledgement slide. This project is a collaboration between with people here, colleagues here at the University of Wisconsin, and in particular Claudia Calderon. She's leading really all the breeding aspect and participatory breeding and interaction with colleagues on the ground, and so Claudia is clearly a major player in this project. But also Sean Kepler and Natalia De Leon in my department of agronomy, who are outstanding corn breeders. We have a great collaborator also at University of Georgia, Jason Wallace. And Jason is, so it's Georgia, it's a slightly different environment, it's south from here. And Jason also has a great expertise in maize microbe interactions. And the project is funded by USDA. Again, it's a public project funded to be public and will stay public. All right, thank you very much for your attention. I would be glad to take your questions.
Thank you very much, Jean-Michel. It was really interesting, a great seminar. So I remind my colleagues that if anyone wants to ask, if you can please write in the chat your question, or even if we are not that many, just put your camera and raise your hand and we can direct the questions. I don't see anyone. Maybe I can start, Jean-Michel. So regarding this last point that you mentioned, the integration and the crossing to try to put the traits in other plants, I have a question there. I mean, because you mentioned the very special kind of traits that there are, right? That they develop the Ariel roots in the adult phase. They produce a very great amount of mucilage, etc. And so if you don't see, or if you don't find this kind of at least some similar traits in other varieties, how, for example, the data that you showed us that Mars was doing these integrations, and you can see some differences in the F1 progeny, but these plants, I'm assuming that they do not have any of these in particular characteristics, or how do you?
So the goal, in fact, of the breeding is to keep the traits we want in these accessions from the Sierra and get rid of those we don't want. So things that we keep in this progeny are the high number of nodes with roots and thick roots. And that's what really we breed for. We breed for plants with many nodes and thick roots. Mm-hmm. And things we get rid of is the size in particular, because really I think what the issue, if people wanted to use these land races, is clearly the size. And also the fact that it's growing over a really long period of time. I mean, it's growing over 8 months or 9 months, 8 or 9 months. So we want to have maize that grows fast, that has a regular size, but that will have many node-free roots and thick roots. And quite frankly, we do see these plants, sort of ideal plants in our progenies. So I'm quite encouraged that it's doable to transfer these traits. But it takes time. It takes time. Breeding takes years and decades.
Okay. Yeah, thank you. So there's some questions now in the chat. So one from Jose Luis Reyes. He's from the scientist from the Institute of Biotechnology that says, can you inoculate other accessions with mucilage and bacteria?
Yeah, inoculation would be inoculation. I guess there you are thinking on the roots, maybe. So what we see, for instance, that if we put that gel, for instance, on the soil, it's degraded very quickly. In fact, we even looked at the soil which is under the roots. So the roots are, I showed you, are dripping with gel, and the gel is sometimes falls on the ground. And so we are wondering, is fixation happening on that soil that contains the gel? And we see almost nothing. The gel is very quickly degraded as soon as it's touching the ground. So part of the trick of the biological system is to keep that gel in the air, on the roots in the air. So it works as long as the gel and these bacteria are on roots that don't touch the soil. So the fact that it's not in contact with the soil is not working. We did a fun experiment just for fun, and it worked, of taking the gel from the corn and then putting plants. It's called brachypodium. It's a small cereal, and we were able to show that we can get nitrogen from nitrogen fixation into brachypodium when brachypodium has its roots in the gel. But it's not practical. I mean, from a so the fact that the gel has to be on the roots in the air is actually part of the system. And unless we have a plant with that kind of trait, it will not work. Yeah.
So perhaps you can see the chat also. Otto Geiger is asking something. Does the aerial root trait get lost when maize is continually cultivated in milpa? This is in association with beans?
I don't know if it's lost. To be honest, I've never, maybe you, Otto, you mean about a long period of time. I was wondering, go ahead.
In evolutionary times.
Yeah, I think that possibly people who did not realize the usefulness of these aerial roots probably got rid of them. And my feeling is that there has been, probably by Europeans, a counter selection against these roots, because when I first started growing that maize here, literally I had two colleagues, I won't say who, in Madison, who told me that I had an ugly corn, that for them it was not a beautiful corn because it has all these roots and the gel. Another colleague came to me saying that I had a fungus on my corn because they were seeing the gel, they were thinking my corn had a disease. And so my feeling is that people, like many people, there is in that culture, probably people did not realize the importance of a gel. And in contrast, local communities, they highly know that a corn with gel and a corn with lots of roots is valuable. So I think that there is a cultural aspect that has been lost with Europeans, and that probably people have selected against these roots. My colleague working on sorghum tells me that for them, when they see many aerial roots, it's not desirable because sorghum for silage, these roots are woody, and so it's decreasing the quality of sorghum for silage in their opinion. But so anyway, my point here, I think that people have lost the knowledge that these roots are useful. Does that help?
Thank you.
So Jose Luis Zúñiga Rodriguez asks, is there someone working on triticale?
So I tried a few collaborations with colleagues working on wheat and progenitors of wheat, and they screened their collections and have never found any wheat or even I look at the brachypodium and we haven't found anything there. Same with in terms of having a same phenotype with aerial roots. We looked at, with the Erie again, rice. We haven't found anything at all. So we think it's in the family that contains maize and sorghum. There is a model there called Setaria. So there are reports of Setaria accessions that have many nodes with roots. I still don't have access, don't have them, but it's in that yeah, we haven't found anything in triticales yet.
Esperanza has a question there. When you measured nitrogen in mucilage, did you remove border cells?
That's a good question. No, we did not. But it's the problem is it's impossible. And I agree that it's possible that some of the nitrogen we see is in fact coming from the border cells, from lysing these cells. But they are so embedded within the mucilage that centrifugation doesn't work. Which, yeah, no. So that's a good question. That no, we don't remove. We did not remove the border cells. And
Then,
if you have good ideas on
thanks for a great seminar. Then Olivia Rojo, well, she says root exudates are essential in specialized association between plants and soil microorganisms and can maintain population of soil microorganisms. Have you observed the effect of other exudates, such as auxins, for example?
Yeah, I think what you're alluding to is the fact that there is probably more than just polysaccharide and simple sugar in these exudates. Well, and yes, and for instance, what we found recently that we had missed until recently is the fact that even you have sucrose in the gel, a lot of sucrose. So the disaccharides, we actually missed them until recently. We also think that organic acids are important. We find actually quite a bit of succinate in the gel, and many nitrogen fixing bacteria love succinate. So the story is more complex than what I presented today and what we published of just sugars and polysaccharides. It's more complex, and so we haven't looked at auxin, or hormones. What we see, but it's not so surprising, is that many of our isolates, bacterial isolates that we isolate from the gel, are auxin producers. But if you also do the same with roots that you find underground associated with plants, you find also many bacteria that produce auxin. But we have not looked at auxin content in the gel. But that's a good idea. But I'm pretty sure that there must be auxin just because we have all these auxin producing bacteria.
Thank you. Another question from Arnaud, that he's from the Biotechnology Institute also. Great talk. Is the mist or climate important for nitrogen fixation with this variety?
Oh yeah, absolutely. Yeah, if you are under low humidity, the plants don't even produce the roots. And so initially my colleagues at UC Davis, who are trying to grow the corn over there, they had not a single Ariel root. The same variety was not even producing roots. So you need to have a good humidity. Here, with what we get in Wisconsin, the plants produce 10 to 12 nodes with roots. So the humidity we get in here is good enough. But you need humidity for the roots to come out. Yes, I know colleagues in Texas also see fewer roots than we do here. So humidity is important, and the gel only comes out after rain. So humidity just in the air is not sufficient for the gel to come out. You need to have actual rain. And the rain frequency is also correlated with how much gel is produced and how much fixation happens. So you need to have humidity and rain. Yes.
Damian from May here from my center did you look at the aerial roots from rice? Is the mucilage and many diazotrophic bacteria?
So I have not found, and if you know some rice varieties that have aerial roots, I have not found any rice variety that has aerial roots, and we did a screen for that. We do find gel produced by underground roots or crown roots even in rice, and the gel composition is very similar. In fact, the gel composition of what you find produced by underground roots of rice, maize, sorghum is very similar. Not identical, but very similar. But it's really a matter of producing, for that system I presented, it's a matter of quantity, and it's a matter of producing that gel outside of the soil.
There's a question from Giovanni Santiago. He says he's from Penn State University. Do you know who is producing the mucilage?
The root cap and border cells, and mostly the border cells. And yeah, I showed some data on the border cells. The border cells from the young roots are bigger and produce even more gel, and young roots have many border cells. So clearly the gel is produced by border cells. And there has been some data also, and we have some preliminary data also on that, of looking at within the gel where the bacteria go, and we see that some bacteria really likes to aggregate around these border cells that produce the mucilage.
Monica Rosenbluth asks, sorghum plants with high amount of aerial roots also grow in places with high humidity?
Yes.
Mazes with less amount of aerial roots do also have nitrogen fixation in the roots?
Yes and yes. So the sorghum that has many nodes with roots and is coming from also tropical regions, and it's places where they have high humidity. Note on the sorghum, I'm not aware that anybody in sorghum was using the trait. You know, clearly people in the Sierra Mije and Totontepec and probably other places in Mexico knew about the trait and of the fact that having many nodes with roots and having gel is good. I'm not aware that anybody in sorghum recognized that in sorghum. Regarding a second question, yes. So if you take the maize varieties that are grown more classically here in the US that have just a few nodes with roots, and if you measure nitrogen fixation in these roots, it's per volume of gel, it's the same nitrogenase activity. But these accessions produce far less gel. So to get 2 milliliters of gel in a tube, we have to go through 100 inbreds called B73 plants to get 2 milliliters, because these plants produce very small amounts of gel, whereas it's just a single root on the giant corn that produces 2 milliliters. So it's really a matter of, honestly, volume and how much gel is produced.
Does mucilage have an effect on root development or other species? Edgar Pascual.
That's a good question. Of other species of plants, I have no idea. I don't know. That's a great question. Again, that mucilage is also produced underground by regular root system, but it's very quickly degraded at the root tips. Do these plants, like dripping with gel, affect other plants? I don't know. Great question. I don't know. Yeah.
So I don't see any other question in the chat. Is there another question? Okay, if not, we thank you really very, very much. Jean-Michel, as you saw, it was an interesting topic for many of our colleagues, and thank you very much for your willingness to present this to us.
Thanks for the invitation. It was a pleasure. And again, if you have ideas, if you want to work together, I'm more than happy to do so.
Okay.
Great.
Thank you.
Bye.
Bye.
Thank you.